Hello Stanley, how are you today?
I have been better, Ronald. I have a little problem.
Yes, I see. You are not wearing clothes at work.
That is not the problem. Today is Friday and I can wear whatever I want on Casual Friday. I have a serious problem.
I will try my best to help. What is your problem?
Do not ask me why, but I have some wastewater and I do not know what to do with it.
Why?
I said do not ask me why.
Oh, okay, well then. You could try and make energy from the wastewater.
I did not know that was possible.
It is very possible. The technology is called microbial fuel cells.
Microbial fuel cells? I never heard of them.
I was researching the topic yesterday and found a great video that can help you.
Really?
Yes. The video was very informative and I learned a lot about poop power.
Who made this video?
A group of Maryland students for ENCE 411.
Thanks. Also, you have a mustache, so I believe you.
Let's watch it.
Let's make a fuel cell. Let's see that one more time. A little slower. A microbial fuel cell uses aerobic and anaerobic reactions to create an electric potential, which can be utilized as a power source. For this project, our microbial fuel cells, or MFCs, consisted of three basic components: a graphite anion, a carbon cloth cathode, and geobacter. In total, 6 MFCs were constructed for this experiment. Geobacter is a particular bacterial strain that is the first organism to oxidize organic matter while using a metal, such as our graphite anion, as its electron acceptor. This graphite anode acts as the electron acceptor of the MFC. As the MFC matures, the geobacter goes through anaerobic respiration on the anode and grows into a biofilm, coating the anode. Since electron transfer is a function of contact between the geobacter and the anode's surface area is a significant variable. The cathode used was carbon cloth with a Teflon coating, making the cathode float on top of the water. This allowed the oxygen in the air to properly diffuse through the cloth and be reduced. The anode and cathodes were then placed in our 2-liter batch reactors. Wastewater obtained from Blue Plains Water Treatment Facility was then used to fill the batch reactors. As the reactors were thoroughly mixed, samples were taken undiluted. Initial COD readings were then taken using the diluted wastewater samples. Following setup, the batch reactors were wired to a multimeter that would measure the electrical potential between anode and cathode. An initial resistance of 50 ohms was applied. Over the course of a week and a half, different levels of resistance were applied and polarization tests performed to obtain an optimal resistance. After setting this resistance, the anodal bacterial growth was measured as a function of current produced. Following the test, final COD measurements were taken and the batch reactors visually inspected. As evidenced by the biolayer growth at the cathode, it was evident that aerobic reduction of oxygen took place.
Wasn't that the best video you have ever seen?
Wow. That video changed my life. The group that made it should definitely get a really good grade.
Yes, it really was the best movie ever. Also Stanley, you do realize that today is Thursday and I am going to have to report you to management.